welcome everyone. Really happy to have this postdoc series from the Indian Society for Developmental Biology. And these are postdocs who are outside on the whole, but it's open to any kind of early career researcher. So if you want to present your work, if you want some feedback on your work, please feel free to sign up. I will now hand over to Bhargav Sanketi. And so I should say that this is this series has been kind of organized by Bhargav, Yamini, and Nikhil. Nikhil is here somewhere. Yamini is in the University of Geneva, Nikhil in Austria, Bhargav is in San Diego. And I'll hand over to Bhargav, who's the MC for today.
Perfect. Thank you, Raj. Thank you, Indulekha. Thank you, everybody, for helping organize this. And welcome, everybody, attending to the first session of Gulmohar. We're a seminar series from the Indian Society of Developmental Biology. And the hope is to highlight early career scientists from across the world to share their work with the Indian research community. And we would like to keep our sessions thematic for the most part. So today we have physics and mechanics of morphogenesis. We will have two speakers. The first speaker is Yamini Ravichandran, a postdoc at the University of Geneva in the RU lab. And she studies mechanical induction of morphogenesis, which she'll be talking about today. The second speaker will be Nimesh Chahare, who's a postdoc at Columbia University in Nandan Nerurkar's lab, and this is in New York. And he'll present about multi-scale dynamics of epithelial sheets. So each speaker will have 25 minutes for the talk, followed by 5 minutes for questioning. And in the end of the seminar, after both the speakers are done, we request both the speakers to kind of stick around on the call. And at this point of time, people will be again free to ask you a few more questions about your science, but also maybe even about your career trajectory or any kind of advice that they're seeking out of you. All right. Yamini, if you're ready, please start.
Yeah, thank you. Thank you, everyone. A big thanks to Rajan, Sonia, and Indu for doing all the vaccines. So I'm a postdoc who's interested in mechanical induction of morphogenesis, and the model organism of choice is hydra, and I will talk a bit about this work that we put out earlier on this year in bioRxiv. So during morphogenesis, self-organization is okay, I just have to move this. Yeah, during morphogenesis, self-organization is a key step that leads to large-scale organization and shape formation. And we observe this in vivo in various model organisms, such as in the Drosophila, gastrulation events, or even in the zebrafish model embryo developmental stages. However, the quest is also to recapitulate these self-organization events, either in vitro or to see the need for these in higher organisms, such as in human embryos. And this is what you can see even when you take embryos that are in vitro fertilized and look at their organization, and you can see that different layers of these embryos, such as the extraembryonic tissue or the embryonic stem cells themselves, self-organize into certain shapes and regions of domains. Yeah. So I study morphogenesis from a regeneration context, and I study self-organization in hydra during its morphogenesis. So why hydra? Hydra is a very simple organism. Simple in the sense that it has a simple body plan. It has an oral end and an aboral end, so one body axis, and it has extraordinary regenerative capacities. So you can study morphogenesis or shape formation from a regeneration context. And how you can follow regeneration, you can chop the animal into bits and it will regenerate. And throughout the talk, I will mostly talk about these large sections. such as the head part, which regenerates a new foot, and then the body column, which can be chopped up and turns into a spheroid, which will then regenerate a new head and a foot. And the foot is capable of regenerating a new head, and that is known as head regeneration. And all the videos of these morphogenetic events are visualized using transgenic life fact expressing animals here, 6 hours post-dissection. So there are videos that are taken over a course of 4 days using a spinning disc, and this is what they look like. So you can see the head that will pulsate, and hydra has this intrinsic ability to pulsate and shape itself. But you can see that due to the presence of tentacles, the animal moves a lot, and it's really hard to even capture it. Whereas the body column here, you can see it will go through the same pulsating movement, but is capable. You're capable of imaging it, and you can see that the new head will form towards the end of the video, and the new feet over there. The same is for the same occurs for the foot, which is going to go ahead and create a new head. So for most of the talk, I will be showing this piece of tissue, which is regenerating a new head, and I'm focused on trying to understand self-organization during this morphogenesis event in hydra. So what are the players involved in self-organization in hydra? So far during this body axis formation event, the self-organizers can be categorized into two categories. First, which is biochemical cues. And these biochemical cues fall under the well-established Spearman and Mangold organizer, which is essential for has been essential for the field of developmental biology. And the same Spearman and Mangold organizer, which was seen in Xenopus, was first described in hydra by Ethel Brown. And the molecules of this organizer comprise of the canonical Wnt pathway. However, in hydra, most of the data on these biochemical cues is static, mostly in situ, immunofluorescence, and so on and so forth. So there was a lack of temporal dynamics of morphogenetic self-organization in hydra regeneration. Whereas more and more studies show with the evolution of physics of life and the importance of mechanical cues, we can, even in hydra is no exception, and we observe studies which show that mechanical cues are important for hydra's regeneration as well. And these studies are first by Albrecht Ott and then by the lab of Caricios, which showed that mechanical oscillations that you saw in the videos of the regenerating tissue are important for regeneration. And then tissue interfacial tension has also been shown to play a role in the sorting of the ectoderm and endoderm in this animal during reaggregation experiments. And lastly, one that was more interesting and inspiring to me was the study which was published in 2021 by the lab of Kinnard-Karen, which showed that basal actin of the animal displayed long-range nematic order, which could drive defect-driven shape generation. I will talk about nematic order and defects later on. And what is most important, even for people trying to study morphogenesis, is that these data give more a dynamic perspective to morphogenesis. They are temporal, and you can actually map the evolution of shape as compared to the static studies that have been previously conducted. So yeah. So in order to understand nematic order, I would have to explain a bit about liquid crystals and topological defects. So liquid crystals are a case of matter where they display long-range order. They have orientational order, so they can reorient themselves and display fluid-like properties. But whereas these particles don't have fixed positional order like solids. Okay, so they're liquid-like and solid-like, and that's why they're known as liquid crystals. But what impacts this in certain aspects impact this long-range order of these liquid crystals is basically what we have on our screens, the liquid crystals on our displays. And what impacts the organization of these oriented particles are aspects such as curvature, topology, and so on. And when they do impact it, what happens is that you have regions of singularities known as topological defects. These regions of singularities look like this, and they have a certain corresponding charge. And these can be calculated basically just by the winding number. So how much you have to reorient them so that they can align themselves along the orientational axis. So these are the topological defects, and they can be asters, vortices, antivortices, comets, and trefoil, and so forth. But what determines the distribution of these topological defects? I mentioned that the arrangement of these defects depend on a phenomenon. Firstly, they have to obey the rules of topology. Topology being, you can deform a surface into any other shape, but not by piercing it or tearing it. And if without piercing it and tearing it, you can deform a cube into a sphere, and it will display the same Euler characteristic, that is the same number of topological defects, and so on and so forth. The only way you can alter the Euler characteristic or a topology of a cube or a sphere is by introducing a hole and creating a torus or a cube with a cavity. Only then the Euler characteristic changes, and the topological defects can now add up to a sum of 0. Okay. If you have questions about this, you can ask me about this. But fundamentally, morphogenesis is a series of deformation events. So if you do not create a tear or a hole, your morphogenetic events will obey the same topological the same rules of topology will apply to your morphogenetic processes. So, and so on and so forth. So that is very important when we are trying to draw from the physics of liquid crystals to describe morphogenesis. So yeah. So enough of all these liquid crystals. Now I come back to the physiological relevance of topological defects and orientational order. So nematic order is exhibited by biological systems. Very obvious in your even in your fingerprints. You can see that you have long range order of these ridges, and in regions where they break symmetry, you will have the appearance of a comet or a trefoil, and so on. But whether they really play a role, whether is it just a correlation or whether it has a role, has been the quest for whether it aids in self-organization or driving forces, has been the quest of biophysicists in the field parallelly using biological systems. So in cell monolayers, a lot of studies have shown that in vitro cell monolayers, cells that have long-range orientational order can display nematic order and can behave like liquid crystals. And a work by Benoit Ledoux showed that cell extrusion events occur. Cell extrusion events in an epithelia occur exactly at sites of topological defects, therefore suggesting that topological defects, these asters and these singularities that I showed, are able of generating stresses which can extrude cells. And cell sorting also is another such event. But what was more fascinating was one study done by in our lab, and which was done by Pau. I think he's in the audience here today. So Pau Guillaume in the RU lab showed that in vitro muscle cells can organize themselves in a circular, if you confine them in a circular micropattern, they can organize themselves into asters and spirals and create three-dimensional protrusions, a bit similar to the cell extrusion event observed in an epithelia, but here describing a 3D process. So that started to describe that topological defects can generate shapes such as even 3D cellular mounts. And then comes the most significant study, which was the in vivo morphogenetic event in hydra coupling actin. So yeah, so in hydra, actin displays liquid crystal nematic order. That is, here you can see a foot tissue where we are looking at actin. We're looking at actin in the ectoderm, and you can see that the actin is absolutely displaying long-range order, and it almost looks like a single cell that is protruding and creating a new, like a philopodia or laminipodia. But actually, it's an entire hydra tissue that is creating a new head and tentacles and so on. And what is even more interesting is what the lab of Kern described, that the topological charge of the topological defects displayed in this actin order add up to 2. That is, the asters correlate with the formation of the new head, which was very striking. And also you can see an aster at the foot, the head, the tentacle, and trefoils that balance out the total charge. And then you have the actin in hydra obeying the laws and maths of topology. But what was left to still understand is that are these actin topological defects that are observed in hydra essential for 3D shape formation, right? At this point, it's still a correlative correlative study. So is it really driving shape formation was what was left to, was the open question in the field. So given the pneumatic interest in the RULAB and my experience with organisms, I started to answer this question in the beginning of my postdoc. So I wanted to alter these actin defects in hydra. And what I wanted to do was, I thought probably if we could deform the tissue, we could give rise to rearrangements of these aster defects, which correlate with the new head formation in the regenerating tissue, or even do something which is harder, but which would be which is very unlikely, is to introduce a tear or a hole into the tissue, and then you can change the topology from a sphere to a torus, and then ask changes in how can changes in topology impact regeneration. Yeah. So first, I sought out by deforming the hydra tissue. Please stop me for give me a heads up like when there's like 10 minutes or something. But yeah, so I'll try to go faster. So to deform the hydra tissue, what I did was I used the foot tissue 6 hours post dissection, and then I subjected it to a soft agro slab compression, and hoping that it will deform the tissue. And then the control scenario that I used was embedding the tissue in agarose so that you can eliminate for effects of agarose and not compress the tissue. To visualize how much the tissue gets deformed, I performed 2-photon microscopy, and here you can see with increasing percentages of agarose how the tissue gets deformed. So with 2-photon, we are able to appreciate the 3-dimensionality of the tissue, and here I render the actin, the magenta being the apical actin, and the blue being the basal actin pneumatic bundles that I described earlier on in my talk. So here in 0.5%, you can see that the lumen is intact, and the animal feels the agarose slab compression on top. Whereas with 1% and 2%, you can almost squish it out like a pancake, and it feels more the compressive stress. And yeah, like 2% is basically like, like a dosa, you're killing it. So after I managed to deform the tissue, now I wanted to observe how this deformation and this compressive stress can impact the regeneration of these tissues. So in order to do that, I followed these compressed tissues under using a spinning disk for 4 days, and I followed how they would regenerate. And the control scenario, not surprisingly, after 4 days, creates a new head, and you can see here it has a degree of freedom, even though it's embedded in agarose, and creates a new head and tentacles that would be visible there. Whereas the agarose compression, not surprising that the 2%, you squish it to death, because that is like the upper limit apparently, and the animal cannot withstand it anymore. Whereas with 0.5% and 1%, we start to observe this unique axis duplication event, where we mechanically induce the appearance of 2 heads biaxial animals, which was never described before due to compression. So actually what you create here is the Lernian Greek hydra, right? Where you chop off the head and it creates 2 heads, which is very, very cool that we previously biochemically you're capable of doing this, but one of the first evidences of showing this mechanically. And now there are more studies showing that confinement indeed plays a role in regeneration. So yeah, then I went on to quantify this and see what is the distribution of these mechanical phenotypes. So all of the embedded agarose tissues are uniaxial, whereas the 0.5% you start to see a large appearance of biaxial animals and a fraction of ectopic tentacle as well. So these are animals which are incapable of creating a new head, but end up just creating like an ectopic tentacle. This is known as a weaker wind phenotype in the field, and with 1%, you start to observe a large population of non-regenerating tissues. That is, tissues that do not complete regeneration even after 4 days. Whereas in wild type scenario, by 4 days you actually do complete regeneration. So it seems like mechanical compression stalls regeneration or slows it down. And yeah, and 2%, mostly the phenotype is that they cannot withstand the weight, and you have a large fraction of death. So then I wanted to understand what determines the distribution of these mechanical phenotypes, like why, a certain percentage of ectopic and a certain percentage of biaxial and so on and so forth. So in order to study that, I wanted to see what is the variability in the assay that I do, because it's just, just compressing with a slab. So where is the variability coming from? And then I observed that when I actually do the squishing, I have no control over the orientation of the tissue under compression, because the tissue is like jelly. It's not very it's not very rigid, let's say, like a mammalian tissue. And so what happens is I'm not able to orient them in a certain order. And these are the three kind of orientations the tissue chooses to orient itself. So what are these? So the lateral orientation being when the tissue is perpendicular to the compressive agarose slab, and the aboral and oral orientation is when the tissue is parallel to the compressive slab. So, and aboral and oral only differs in the polarity of the tissue. So one where the head faces the agarose and the foot side faces the agarose. And this is what they look like in terms of actin orientation. So you can understand why logically the animal would prefer this, because it's easier to withstand the weight, and it can regenerate like this. Whereas it would be very hard for it to push against the agarose and regenerate. So to see how much the animals choose to orient themselves in this orientation, I quantified it. So at T6 hours, I saw how they were distributed. So at 6 hours, in 0.5%, where the agarose is very slurry and soft, the animal is capable of reorienting itself laterally, and almost all the animals choose the lateral orientation. Whereas 1% and 2% is stiffer agarose, and the animal fails to reorient itself. And you start to see evidences of aboral and oral orientation highlighted here, which is the same with 2%. So yeah, indeed, there is a certain population. And to see how this orientation impacts the regenerative phenotype at day 4, I followed these animals over the course of 4 days and saw what they turned into. So, so 0.5%, all of them are lateral, and this is how all the lateral tissues regenerate after 4 days of regeneration and confinement release. You start to see that you have the biaxial, ectopic, and this is how it's distributed. Whereas in 1%, oral and aboral are least preferred, and this is how they look like. Whereas lateral, very interestingly, absolutely phenocopies 0.5%, showing that it is not the percentage of agarose that is important, but what is important is the orientation of the tissue, which dictates this biaxial phenotype. And this was very striking and interesting to identify. And lastly, 2% just shows that it's the upper limit, and most of them die. So orientation you can't even determine effects based on tissue orientation. Coupled with mathematical modeling, which I don't show here for the sake of time, we were able to then show that indeed topological defects are, here is just a correlation. We show that multiple topological are essential for shape changes, and we show that multiple Astor defects correlate with multiple heads. So such as 2 heads in the lateral compression state create 2 Astors, and you have that's why we have the multiple head formation. But then I was still wondering that there is a large proportion of tissues that.
Sorry.
Okay.
5 minutes.
Yeah. I'm almost done. So how about the actin orientation of tissues that do not complete regeneration even after the course of 4 days? And that is the no regeneration tissue population that I showed you earlier on. So these tissues, these are the tissues that I'm talking about. After 4 days, in 1%, there's a large population that does not regenerate. So I wanted to see why that doesn't happen. And, if I release the compressive stress, whether it will regenerate, because hydra is destined to regenerate. So first I wanted to look at the actin orientation and see what was causing this. So the actin orientation of these tissues looked intact. There's long-range actin order, but the only thing is that they fail to break symmetry even after 4 days. So I wanted to release these tissues and see whether they sort themselves into biaxial, ectopic, and uniaxial hydra. And indeed, post-confinement release, a large chunk of these tissues do regenerate, whereas a smaller fraction still does not persistently regenerate. And when I paid attention to these small fractions that persistently do not regenerate, I observed that indeed orientation plays a role here again, and most of them are aborally oriented, parallel to the compressive stress like this. So here we're looking at the foot tissue of hydra that is regenerating. And you can see that now the oscillations which were going like this have been changed to radial oscillations like this due to the compression. And what has happened is you create a tear. You create a tissue tear, and thanks to hydra's extraordinary regenerative property, the tissue has healed, and what we have now is a donut tissue. And if you remember in the beginning of my talk, I told you this is a unique change in this is an induction in a change in topology, because we have gone from a sphere to a donut. And for like biologists like me, this is like knocking out topological defects, because if there is a torus, then if it's perfectly symmetric, then it's the only combination where you can eliminate these aster-like topological defects that I was talking about. So indeed, I wanted to follow the fate of this torus and see whether the lack of topological defects impacted its regeneration. So to appreciate the 3D morphology of this tissue, after releasing it, I can see that indeed it is toroidal and has a perfectly symmetric actin, as you can see here with the light sheet image, and displays curvature phenomenon such that of a torus. And following it in the spinning disk for a couple of days, you can see here that it keeps going on oscillating, but never establishes a new head or a foot, which is striking, because for a hydra, a week later, having intact tissue and having all the biochemical gradients, you don't create a new head or a foot is really bizarre. So what I've managed to do here is get the animal stuck in a math puzzle, and it fails to break apart due to the lack of topological defects. So yeah, with that, I would like to summarize. And yeah, please have a look at the paper. It will do a better job than me trying to summarize what I've shown. But I hope I try to deliver the message that that topological defects correlated with heads, and we induced more topological defects and proved that by showing that you can have two actin defects that can correlate with two heads, and then you can knock out these topological defects in a toroidal shape, in a defectless torus, and then you can stall regeneration even for a week, which is very unlikely for hydra. So that was the null hypothesis. And then you can rescue this torus by creating a toroidal hydra, which is capable of generating defects and then creating a head. So yeah, and that is the toroidal hydra. So yeah, so with these three phenomena, then we can now say that topological defects are indeed essential for shape generation and can be characterized as a mechanical organizer driving possibly the self-organization of biochemical players that we do not know of at this point in the animal. So yeah. With that, I'd like to thank the RU Lab. This is Aurélien. Thanks to him for letting me do crazy stuff with the hydra. And then I would like to thank Matthias Vogt from the lab of Brigitte Galliot, who was very instrumental in the beginning of this project and who taught me a lot about hydra. And Dan, who's the theoretician for this project, who did a lot of mathematical modeling to recapitulate the curvature deformation events coupled with the topological defects. If you're interested, you can see his modeling in the paper. And yeah, and Carsten Kruse, who was also the theoretician. And thanks to the Hydra, and thank you for your attention. And thank you to the Indian Society of Developmental Biology for hosting a great meeting earlier on this year, where I met a lot of wonderful people, and for this invitation. Thank you.
Thank you, Yamini. I think you have a question in the chat.
Yes. I can read out the first question for you. So, how does the compression alter the topological defects? If it's uniform compression throughout the animal, will it alter the defect? And this is from Upal Chatterjee.
Okay. So the goal is that it doesn't really the compression doesn't directly alter the topological defect, right? It doesn't directly alter it. The goal is that the compression changes the geometry of the tissue and keeps it confined in a certain phase, a certain way. So then it gives rise to singularities in regions which would not be possible before. For example, if you have a uniaxial hydra, then it's a cylinder. It doesn't feel any compression, so it's a cylinder like this, and it's going to create its new +1 aster here. But if you take this cone and you squish it, so what happens is you create two regions of weaknesses where you can create the two +1 asters. So that is the thinking behind it, that when you deform it, you change the geometry and you give rise to regions of singularities in different areas of the tissue. Yep.
The next question is from Meghana GC. Meghana, if you would like, you could unmute and ask the question yourself too. Okay. I'll do it for her.
Yeah.
So, would there be a possibility of head regeneration in toroids by adding drugs like ALP and ALZ, which I don't know what they mean, which
Yeah, I know that.
transiently activates Wnt?
Yeah, that's a cool question. That's what I would like to do actually. But the problem is that well, not really problem. The bottleneck is that I'm not able to mass produce them. I'm currently working on mass producing the donuts. So yeah, so once I manage to mass produce the donuts, I'm going to do things like this. Basically, yeah. She's absolutely right. I plan to do that. Yeah. And I hope we can
perhaps
rescue
have
these donuts. Yeah,
time for
sorry.
one final question. Anubhav, would you like to ask it in person? Okay. Does this mechanical perturbation redistribute biochemical cues, especially one that regulates axis formation? Is the question from Anubhav.
I do think it does, but the problem is that we cannot visualize biochemical, and that's what I started off with in the beginning of my talk. We cannot visualize these. We have reporter lines, so I'm looking at the reporter of Wnt expression and beta-catenin and SP5 at the moment. So seeing how just reporter lines can change. But I absolutely think, because there is this thinking of reaction diffusion mechanisms, right, in development, which is one of the key things. So in hydra also there is this thinking that there is a graded element, and of course if we compress it, the graded element will get perturbed. So, but I do not know of this. I have only mapped the actin, and I've only mapped the actin, and I've only studied the actin organization. But there is a whole a line of research which has to be done under compression and change of biochemical fate. Yeah.
Now, so if anybody has any last questions, I know Raghavan has asked that he has a question in the chat, so you can go ahead and ask.
Yeah, it was cool to see these defects being like localizations of future developmental points. But what do you think is leading to these defects, or what is actually positioning these defects in the right place?
Yeah, that's a good question. I think, at least in the regenerative context, probably the matrix has to play a role, because instabilities in the matrix would drive something. And also, with the compression scenario, it's the compression that dictates the geometry and then localizes defects in regions where you're having, bends and curves and so on and so forth. But if you ask me what is physiologically the primary, like for example, during embryonic development, how the actin sorts itself and why it localizes one defect here and one defect there, I don't know. This we don't know yet, and I think the field does not know yet. And we need to go and look at the embryo, for example, where there is de novo actin organization from disordered disordered actin to order, and then we see it. And I think there we can get clues. And there, I think, yeah, there we have to see. Yeah, there has to be some activity driving this, right? Because even if it is a pneumatic system, it's an active system, so probably myosin or something, or these kind of activity driving players should be positioning defects. Would be my hypothesis, but yeah.
Yeah, because I was thinking it could be the length of actin, because you could see that in the branched form, you have actually the length of the actin segments are changing as they pass through this curvature, right? So this is one thing. And also in your arboreal phenotype, so you also have these actin filaments of almost like constant length, and they don't form any defects, right? So yeah, I was just thinking on this line.
Yeah, but the arboreal type, actually, the problem is you seal it and you trap it. So in a torus, you have the only region of disorder is forming in the center where the tear is occurring, right? So when this tear has occurred, unfortunately, fortunately, the animal prefers to first heal itself. So it because it's always programmed to heal wounds. So it immediately heals its wound before it thinks about creating a head. But I didn't show it in my slides. How we actually rescue a toroid is by taking advantage of a spheroid, which is the body column of the animal, and the body column of the animal under compression, it undergoes disordered actin phase, and then orders itself, and then creates the spheroid, and then creates the new head and the foot. So what happens is I take advantage of that disorganization, and then I compress it. So when the compression happens, the tear occurs in a disordered actin background. So what happens is that while the tear heals, the disordered actin creates a +1 aster. So it's not topology, but it's the ability to create a topological defect that you can then create a toroidal hydra. So yeah, in the defectless torus, I don't think actin's persistent length is being the problem to creating a topological defect. In the defectless torus, the problem is that actin is too ordered. The system is extremely ordered to break symmetry anywhere. Yeah.
Okay. Yeah. Yeah. And one last question. So do you also see these kind of actin arrangements on both sides? Like because you have like two layers of epithelial tissue, like cells, right? In hydra. Do you also see on both sides, or?
So you're talking about the ectoderm and the endoderm, right? So in the hydra, yeah. So everything I've shown is in the ectoderm, where actin is parallel to the body axis, and we're looking at this. So Kinneret Karen also has mapped pneumatic order in the ectoderm and so on and so forth. And on the endoderm, first of all, for technical reasons, it's just hard to visualize depth, for tissue depth, right? You can visualize it. I have videos of the ectoderm. And also what happens is in the ectoderm, there is less apical actin. So everything that I'm showing is basal actin bundles, but there is apical actin in the cortex also. So then you can start to see the cell periphery. So I know that in the lab of Kinneret, for example, they do a lot of cell segmentation to separate these two. But thanks to my compression setup, the basal actin comes very close to the field of view that you can actually visualize the basal actin more prominently, and you don't have to segment out the apical actin. But I'm going away from your question. So, but the endoderm as well displays pneumatic order, is just perpendicular. There are mirror defects. So
I see. Yeah.
your +1 aster, in the region of +1 asters, you start to see vortices, because the endoderm is circumferential, and actually you need this crisscross actin kind of actomizing kind of organization, so that the animal can go through its muscular activity, and this is driving the oscillation, the fundamental oscillation that the animal does to eat, to digest, to puke, to move, and stuff like that. So yeah, so there are mirror defects and they obey pneumatics. But if you ask me whether I visualized it, I've not visualized it, because there is no double actin line. We would like to create one, and that would be nice.
Yeah, very
So we
cool.
can only
Very cool
visualize.
work. Yeah.
Thank you.